Okay, so I'm gonna talk about today about the Convit Vax by the Foundation of Jacinto Convit Organization. First of all, I'm gonna present what I'm going to do with the steps. So I'm gonna begin with a little introduction, a recapitulation about what we have seen already about the vaccine, the objectives of today's presentations, the procedures and methods, following by the experiments and their interpretation, and then a quick summary. So first of all, I wanted to add a little quote. Breast cancer changes you and the change can be beautiful. This is by Jane Crook. And with the help of Convit Organization and their various studies conducting the utility and benefits of their vaccine, so these changes can be made possible for many, many other women struggling with breast cancer. So we already saw in the last presentation and the last researches that the BCG vaccine, along with the formalin and the homologous tumor cells, present very immune benefits for the breast cancer. They present a robust immune system. They fight against these Bragg's and Treg cells. They help with the proliferation of neutrophils and B cells. But we didn't see and we didn't test in the last experiment if there was any side effects to this vaccine. So why not test it on normal mice models to see any unwanted side effects? All along these experiments, we wanted to see if there's any toxicity for this vaccine, if there was any weight loss, appearances, morphological differences. We did some sample runs, urine sample runs, and we examined also the spontaneous behaviors. So same thing as the first presentation, we did different treatments along with a control group. So we divided the normal mice into 6 groups. The first group was a control group stored in BPS. The second one contained the homogeneous tumor cells. The third group contained the BCG, the fourth the formalin, the fifth the homogeneous plus formalin plus BCG. And the difference between the 5th group and the 6th group was that it's the same vaccine, but it was more concentrated, like 3 times more. So before we begin the experiments, about how we proceeded a little bit. So the procedures were safe. We used a low concentration of formalin so that we can proceed safely, and so that the bacteria and the BCG were not alive and did not interfere in the experiments. We of course had filtered air, we had a constant temperature, and of course the animals and the mice were given constant food and access to water. And other than that, we also had like a relative humidity of 40 and 70%. We injected a volume of 100 micrometers of the vaccine. For the homogeneous solution, how we did get it, we injected a tumor in these bulb C mice, and when the tumor reached 2.5 centimeters, we used propofol, and then we euthanized these mice, so we got the tumor cells. After that, when we injected these doses on the mice, we looked over after 4 hours, 44 hours, 48 hours after each dose, and we did some analysis, histopathological analysis, so the analysis of the tissues through H&E stains. So now the experiments. We did 4 different experiments testing 4 different things for the side effects of these vaccination. So first of all, for the first test. Okay, for the first test, we tested the general deficit and weight in grams of these mice. And for the general deficit, we usually measured like the eyes, the fur, the posture, and all that. And the scores were going from 0, so this means no general deficit, to 28, this means general deficit. Concerning the body weight, there was no significant differences between all the groups and the control group that didn't receive the vaccine. But concerning the general deficit, there was also no changes, like every group had a 0 general deficit, but G6, the group which had a concentrated solution of the vaccine, had a little general deficit, but it's not that big of a difference. So for concerning the test number 2, we evaluated the site of the injection, and we saw if there's any redness, if there's any bumps, if there's any modification for the mice. And we saw, as seen in the picture D, that the group G3, G5, and G6, which received the BCG concerning G3 and the vaccine concerning G5 and G6, received a little, had a little bump and redness, which is the technical word for it is, I guess, erythema and papule. So they had those, but it went away after a bit and it wasn't that serious. So concerning the test number 3, we did some urine tests, some blood tests, and we also saw the creatine and A+ level. Concerning these can also affect like the energy and the functioning of the muscles of the mice. So concerning the urine tests, we got them, it was determined at the day 28, and we made a single mouse like urinate in a plastic wrap. And of course, we got the results by microscopy. We saw that the pH level, the density, the leukocytes, proteins, glucose levels were all the same compared to the control group, which didn't receive the vaccine. So there was no change in the urine tests. Concerning the blood tests, we also saw no change, just a slight decrease in just a slight decrease in I'm sorry, wait. In just a slight decrease in mononuclear leukocytes compared to the increase in polymorphonuclear leukocytes. For the clinical chemistry analysis in mice models, we also saw no change in creatine levels, in Na+ levels, and all that. For the test number 4, we wanted to focus if there's any necropsy in the results, if there's any morphological differences and histopathological differences also. So everything went normal except for the thymus in G2 and the lung in G4. They showed a little increase in weight compared to the G1, compared to the control group. But there was no apoptosis viewable. There was no edema. The kidneys were great. The livers were great. Also, the capillary were good, and there was no problem with breathing. And we know that because there was an unchanged pulmonary parenchyma, and the pulmonary vessels showed intact endothelium. So through H&E stains, we saw that the Convitvax had no effect on organs. And also concerning the morphological differences, there was no morphological differences that was that significant. So to interpret everything and then also summarize, for the observation of the vaccine, we saw no death, of course. There was no deteriorated conditions. Most mice had normal general deficit scores. Only group 6 showed a little general deficit score. Concerning the brain and weight analysis, we had a normal weight and structure. And if there was any differences, it was possibly related to neurological involvement. There was no pathological changes in urine and no necropsy. For the blood analysis, we saw no toxicity and the parameters were normal. And finally, for organ functionality, there was no effects on them. So for the summary, the ConvitVax showed no toxicity towards healthy cells and a safe approach for breast cancer immunotherapy. Thank you.